Hi, I'm Mary McQuilkin, adjunct faculty for the University of Alaska Fairbanks Center for One Health Research. This lecture is divided into two parts because I realized I can't just jump right into talking about vector-borne and zoonotic diseases without first going over the terminology used in epidemiology to describe diseases and disease transmission, so that's part one of the video. Part two continues with more examples of zoonotic and vector-borne diseases, including some that pose unique threats to the health of humans, animals, and ecosystems in Alaska and the circumpolar north. The term endemic refers to a disease that's usually present within a geographic area. It's important to remember that diseases have natural variation, which you need to consider when determining what qualifies as an epidemic, meaning the disease is clearly in excess from what would be normal. The diagram on the right shows the normal fluctuations in an endemic disease, and then when the disease is clearly in excess of what would be expected, you see the epidemic. We're all very familiar with the COVID-19 pandemic, which is an example of an epidemic that became worldwide. Other historical examples include the Spanish flu of 1918 and H1N1 in 2009. An example of something that's endemic is anthrax in hippos in some parts of Uganda. In Queen Elizabeth National Park, the hippos will sometimes get sick from anthrax that naturally occurs in their freshwater habitats. So the hippos are just going about their normal business, eating algae in freshwater lakes, and sometimes they do get sick and even die of anthrax. But the anthrax is always there. It's naturally occurring in that environment, so it would be considered endemic. But if someone was to take the anthrax and make it into a powder, like the anthrax scares back in 2001, they could send it in the mail potentially and use it as a bioterrorism agent and perhaps cause a epidemic by introducing anthrax outside of its normal environment. This shows when H1N1 exceeded the seasonal curve, making it an epidemic. You can see that nice normal variation up and down every year, and then after the 2009 season, it just goes clearly in excess of what would normally be expected. There's many ways disease can spread, and a particular pathogen may have one or several modes of transmission. Horizontal transmission consists of common vehicle, which can be single, multiple, or continuous exposure, contact, including person-to-person transmission, or through a vector. Vertical transmission, also known as mother-to-child transmission, spreads disease from one generation to the next. Epidemiologic curves, also known as epidemic curves, are typically displayed as a histogram of bars with time on the x-axis and cases on the y-axis. The little green drawings across the bottom are simplified versions of epidemiologic curves because I wanted to introduce the basic shapes of epidemiologic curves before getting into the detail. It's helpful to understand the gist of what you would be expecting with different types of transmission, because a lot of the time when you look at real data, it's not ever going to look exactly like any of these, but it can look close enough that you can get an idea of how an infection might be spreading by the shape that the cases are making on the epidemiologic curve. If an outbreak is a single exposure, you'll typically see cases rise rapidly to a peak and then fall gradually. And most cases will occur within one incubation period of the disease. If you see this pattern, but there's a lot of outliers, such as a second smaller peak forming, that could be from secondary transmission, which gives you a clue that perhaps the original index cases are spreading the infection to household contacts, or perhaps it was a single place the infection was coming from and a second group of people got exposed after the first group. An intermittent exposure will look similar to a continuous exposure, but it'll usually make multiple peaks, and the length won't have any relation to the incubation period because it's based on when people were exposed rather than the course of the illness. An example of an intermittent exposure would be if a contaminated food product was sold over a period of time or perhaps served at a cafeteria over a period of time. Like if Taco Tuesday was contaminated, every week you would get a new spike, and it would keep happening regardless of how long the incubation period of the infectious agent is. With a continuous exposure, the number of cases won't go down because it's some sort of factor that the people can't get away from, so they're just continuing to get exposed. With a continuous common source, cases increase gradually and may plateau, and it will last for more than one incubation period. And with person-to-person transmission, you tend to see this spiky kind of shark tooth pattern as the cases are going up and going down. If bear meat contaminated with trichinolosis is served at a large family gathering, how would you expect the epidemiologic curve to appear? It would most likely look like a single exposure because everybody's going to eat it at that one meal, so you wouldn't expect there to be additional spikes in cases after that. Everybody should start to get sick around the same time, and then you shouldn't get more people getting sick from that one exposure afterwards. Following a severe storm, which caused septic systems to overflow, a village's drinking water is contaminated with E. coli. Residents have no other drinking water source, but government and tribal personnel are overwhelmed coping with other damage caused by the storm. How would you expect that epidemiologic curve to appear? This would likely be a continuous exposure because in this example, people don't have any other source of drinking water. So unless people had stockpiled bottled water, they probably would just keep getting exposed because they wouldn't have any way of avoiding it. A person infected with SARS-CoV-2 travels to a rural village. The infection quickly spreads at the church, school, and local store. How would you expect this one to look? Person to person. So we'd see the spiky appearance, typically increasing at peaks one incubation period apart, which would increase in magnitude as it spreads person to person. If you look at early COVID-19 data, that was a great example of this, where you could see really clearly that pattern where people were spreading it person to person with each wave. So you got this jagged line as the cases were increasing and then as they decreased again. Measles is an example of a virus that is much more contagious than COVID-19. It's transmitted through infectious droplets or by airborne transmission. I mentioned my job working for the CDC quarantine station in San Francisco in a previous class. And for reference, if someone is on a plane with measles and is infectious, they have to do a contact investigation for the entire airplane because it spreads so easily. Even with the filtration systems, everyone on board would be at risk of getting measles. Fortunately, we have a very effective vaccine. The measles, mumps, and rubella vaccine was licensed in 1963, and the 2-dose series is 93% effective, which is really great. You can see on the graph of reported annual rate of new cases and deaths from measles per 100,000 people in the population, the sharp decline after 1963 thanks to the vaccine. This graph stops after 2015, but global cases of measles increased by 18% from 2021 to 2022, which has been attributed to a decrease in vaccinations in recent years. During the COVID-19 pandemic, many people were afraid to visit primary care offices and pediatricians' offices, which contributed to children missing or delaying their vaccinations. And after COVID-19 vaccines became politicized, this contributed even more to a decrease in confidence in vaccines overall. The MMR vaccine was the original target of vaccine misinformation. There was a paper published in The Lancet by Andrew Wakefield back in 1998, which claimed that the MMR vaccine caused autism. The paper was an uncontrolled case series with a sample size of only 12 people, yet it received wide publicity. A correction was published almost immediately, and then the entire paper was later retracted. Shortly after it was published, 24 epidemiologic studies were conducted that demonstrated there's no association between MMR and autism. But unfortunately, the paper received a lot of press, and even though it was retracted, it has been cited by many people promoting vaccine misinformation for decades. Another factor is that because the MMR vaccine was so successful, most people who weren't alive in 1963 don't remember these diseases, so people have forgotten how serious they were before vaccines were widely available. Young parents deciding whether or not to vaccinate their babies for measles, mumps, and rubella may have access to misinformation on social media, but they may not know anyone who has ever seen these diseases or experienced them firsthand. Unfortunately, 1 in 5 people in the United States who contract measles require hospitalization, and measles can cause serious complications, including pneumonia and encephalitis, especially in infants and children under age 5, and it can be fatal. I brought up measles to segue into introducing the concept of herd immunity, which often gets misunderstood. Herd immunity is the resistance of a population to attack by a pathogen to which a large portion of the individuals in the population are immune, thus lessening the likelihood of a person infected with the pathogen of coming in contact with a susceptible individual. If the majority of a population, the herd, is protected, such as through immunization, then the risk of an epidemic is low. Let's say Jane gets infected with measles at her dance class. Then she goes to school and plays with other kids. In Community 1, 6% of the kids are susceptible to measles. Jane comes into contact and exposes 3 other kids to measles. The kids who are yellow are susceptible to measles, but the kids who are black are protected or immune, such as through vaccination. All 3 kids that Jane exposed didn't actually get measles because they were protected. In Community 2, 50% of the kids are susceptible to measles. Like before, Jane exposes 3 other kids, but this time, 1 out of the 3 was susceptible to measles. And that kid subsequently comes into contact with 4 other kids and transmits measles to 2 more kids. You can see following the little arrows representing exposure to the previous person who was a case of measles, how easily an infection will spread if 50% of the community is susceptible. In Community 2, most of the vulnerable students would become infected because there isn't herd immunity. The percentage of protected individuals in a population needed to achieve herd immunity differs for each pathogen. For measles, the herd immunity threshold is about 95%. So even if 5% of kids either don't get vaccinated or can't respond effectively to a vaccine because they're immunocompromised, or if it isn't safe for those kids to get the vaccine because of their medical conditions, it would still be okay. We can have 5% of the population not get vaccinated and still prevent the spread of measles because of herd immunity. This diagram compares the incubation period for different foodborne illnesses, but it's not an inclusive list. It's important to know the incubation period because if you know the agent and you know when people got sick, you can work backwards to figure out what the exposure was. For example, if someone involved in an outbreak tests positive for hepatitis A, we know the incubation period is 1 month, so we can then ask the person about possible exposures 1 month prior to illness onset. If the agent remains unknown, but you know the exposure and the onset of illness, these 2 data points can help you identify the causative agent. Window period is the time from infection to when the infection can be detected with testing, and diseases can typically be transmitted during the window period. For example, with most past COVID-19 variants, people were able to transmit the infection before any test could detect their infection. Period of communicability is when a person can transmit the infection, including carrier states. Typhoid Mary is a classic historical example of someone who transmitted typhoid to many people over time. She was an asymptomatic carrier of Salmonella typhi, which causes typhoid fever. So she didn't think she was sick, even though she was able to spread the infection. She was a cook by trade. Once she was identified as a carrier, public health authorities told her she could not continue to work as a cook but unfortunately she did anyway and infected well over 100 people with typhoid fever. Virulence is the proportion of people infected who become seriously ill or die. As you likely remember, during the COVID-19 pandemic, the big concern that most people in public health and related fields were watching for was a variant that would be more virulent. The Delta variant was certainly concerning in that regard, but fortunately, over time, the virulence of SARS-CoV-2 variants has decreased. So, a smaller proportion of the people who are infected become seriously ill or die. The epidemiologic triangle is a way of describing disease transmission by classifying the different variables involved. This isn't an exact science or anything, it's just to help you conceptualize how a disease is being transmitted. It's not always clear cut how something should be classified, and there's cases where a certain thing could be classified differently for a different disease. The host is an organism that harbors the disease, usually humans or animals, and sometimes a host will get sick, but that isn't necessary for something to be considered the host for a pathogen. Host factors can also include whether someone is immunocompromised or has a condition that increases their risk of infection. For other conditions, epidemiologists consider host factors such as occupation, cultural factors, genetics, household size, or lifestyle. A vector can be a mosquito, a tick, or any other living thing that transfers the agent to the host. I'm using what some would consider a very broad definition of a vector here. Some define vectors in terms of an infectious agent's importance to human health, for example, but I think these more narrow definitions are counter to One Health because they're rooted in a perspective that prioritizes humans above all else. I prefer a broad definition that can be applied to plant pathogens or non-zoonotic animal diseases, because we can't presume to know the true importance of a disease just because it doesn't seem to impact humans directly. A broad definition of a vector does risk oversimplification, so it's important to remember that every disease's life cycle is unique, and the nuance of how each variable interacts as part of the system should always be considered. An agent is what causes the disease. An agent can be biological, like bacteria or plasmodium, or it could be chemical, like a poison, or physical, like radiation, or it could be nutritional or lack of nutrition. Other examples of agents include smoke causing lung injury or a motor vehicle accident causing physical injury, and an environment can either be conducive to transmission or it may impede transmission. For example, a warm climate that has lots of standing water collecting in buckets and old tires and ponds would be conducive to transmission of malaria or other diseases for which mosquitoes are the vector, but a really dry or really cold environment would prevent mosquito-borne illness. In epidemiology, we can intervene at any point in this triangle. So it can be helpful to frame a disease like this when thinking about what type of intervention might be most helpful. In the example of Schistosomiasis, which is endemic to all freshwater in Uganda, tiny cercariae, which are depicted on the lower left over agent in blue, break free from freshwater snails after being produced in large numbers within the snail, and they're able to swim through the fresh water to find a person, and they can penetrate human skin. They can also go through other epithelial surfaces such as the mouth or throat to gain entrance into their host. The definitive host for schistosomiasis, or the intended host, is normally a bird, but once they get into any suitable host, they will migrate through the vascular system of the animal until they become sexually mature and produce eggs, which are then passed in the waste of the animal, be it a human or a bird. And once in the water, they find freshwater snails so they can continue their life cycle. Swimming in fresh water in endemic areas would be considered a behavioral risk factor for schistosomiasis. Note that when using this paradigm, things aren't necessarily always in the same role. Humans can be a vector for some infections. For example, if hikers spread non-native algae on their boots from one hiking trail to another, In that example, the humans are transferring the algae, which is the agent, between different host environments, so the human would be the vector. Here's a practice question. The Zika virus is transmitted by the Aedes aegypti mosquito. In this case, the mosquito is the blank, and the virus is the blank. The mosquito is the vector, and the virus is the agent. Again, a vector is the living thing that transfers the agent to the host, and the agent is what causes disease. Here's an example that doesn't fit easily into the Epi Triangle format. Toxoplasmosis is a zoonotic disease caused by the protozoan intracellular parasite Toxoplasma gondii. Cats are the definitive host and shed oocysts in their feces. Toxoplasmosis usually doesn't cause symptoms in humans, but it can be dangerous for immunocompromised people, such as people who have cancer, HIV, or are pregnant. Infection during pregnancy can result in vertical transmission and miscarriage. If a cat has toxoplasmosis, the oocysts take about 24 hours to sporulate and become infectious to humans, so pregnant cat owners can reduce their risk by diligently cleaning the litter box, wearing gloves and washing their hands when coming into contact with cat waste, or by having someone else who isn't pregnant clean the litter box. But most human infections aren't caused by direct contact with litter boxes. Transmission can occur through food and water contaminated by cat feces, so people can get toxoplasmosis by ingesting unwashed fruits and vegetables, or more commonly, they can become infected indirectly when farm animals are infected because their food or water was contaminated. For example, a cow grazing in a field contaminated by cat feces ingests oocysts on the grass, then the cysts mature in their muscle tissue, which end up in your hamburger. You could also be infected from a cow or goat by drinking unpasteurized milk. Like farm animals, marine mammals can also serve as intermediate hosts for toxoplasmosis because they can become infected via contaminated seawater, then cysts mature in their tissues and would subsequently infect humans if consumed without adequate cooking. Humans can be classified as incidental hosts because they aren't the intended host of the agent. Rodents are an intermediate host that plays an important role in perpetuating toxoplasmosis by eating or coming in contact with infected cat feces. Then the infected rodent gets eaten by another cat, who then gets infected. When the parasite infects a host, it targets cells in the muscles, liver, and nerve tissue, including the brain. Mice lose their innate fear of cats when enough of their brain cells have become infected, increasing the likelihood they'll get eaten by a cat. In this way, by damaging rodents' brains, toxoplasma helps perpetuate its own existence. Cats are the only animals that pass oocysts in their feces. They're the only ones capable of helping toxoplasma complete its life cycle. Humans and other animals like cows are incidental hosts because we aren't required for toxoplasma to complete its life cycle, and we don't make a significant contribution to the parasite's persistence. Most people don't infect anyone else, and even if a cow infects many people who eat it, that doesn't help the parasite much either, since both humans and cows don't shed oocysts in our feces to infect more cats. Could rodents be considered a vector for toxoplasmosis? Yeah, using the more broad definition I cited previously in this lecture, rodents could be considered both an intermediate host and a vector. When they get infected and then get eaten by a healthy cat, they perpetuate the Toxoplasma gondii life cycle. In their role, they're transferring the agent from the environment to a host. Zoonotic diseases are those transmitted from animals to humans either directly or indirectly. Many of these pathogens can also be transmitted from humans to animals. So, for veterinarians and those applying a One Health framework to understand how a disease impacts an ecosystem, a bidirectional definition is likely more helpful. Rabies, brucellosis, anthrax, and COVID-19 are all examples of non-vector-borne zoonotic diseases. Diseases in this category can be transmitted directly through animal contact, such as bites or scratches, or indirectly through contact with animal waste or contaminated environments, rather than a living vector. Previously, I mentioned a more broad definition of vector can be helpful when applying a One Health perspective to threats to animal, plant, and ecosystem health. But arthropods such as mosquitoes, fleas, and ticks are classic vectors when the focus is human disease. Zika, malaria, dengue, and chikungunya are examples of vector-borne diseases that are not zoonotic. Diseases in this category are transmitted by vectors such as mosquitoes, fleas, or ticks, but the primary reservoir for the pathogen is humans rather than a non-human animal. This is in contrast to zoonotic diseases that require an animal reservoir, like a rodent for plague, to maintain the pathogen in nature. Plague, Lyme disease, yellow fever, and West Nile virus are all examples of diseases that are both vector-borne and zoonotic. Vector-borne diseases account for more than 17% of all infectious diseases and cause more than 700,000 deaths annually. Cases of vector-borne diseases have increased significantly in the past couple decades. In the United States, more than 1 million cases of vector-borne diseases were reported between 2001 and 2023, and the annual reported cases more than doubled during this time period. This is not the end of the lecture. Continue watching by clicking on Part 2 for more examples of zoonotic and vector-borne diseases, including some that pose unique threats to the health of humans, animals, and ecosystems in Alaska and the circumpolar north.